thank you for the invitation. It's always a pleasure to come back home for professional reasons and add to that some personal enjoyment. We're also working in the neoantigen field, and I would like to show you. So we're a lot of us working on it. So we're trying to approach it in a way whether we can improve the way we detect neoantigens, and I would like to show you a bit of that. These are my disclosures. So I would give a brief introduction, but I think with both previous talks, I won't have to spend too much time on it. But I'll start with this slide on where we question ourselves whether there is a role for the immune system and for immunotherapy in dealing with cancer cells, because you see the peak of incidence of cancer occurs after reproductive age, so you can question whether there was a selective pressure for the immune system to deal with cancer cells. But we also know for a long time that immune-suppressed people or people taking immune suppressants at higher risk for development of cancer. It has to be said that a great deal of these are viral-related, but there are also some cancers that are not viral-related that appear in this type of patients. Thank you. The pointer works, but the... how do immune cells detect cancer cells? Well, there are several ways of that happening. One of those is related to the response to stress signals by the cells themselves. So if you have DNA damage or if you have activation of oncogenic pathways, cells may then induce the expression of certain ligands, and these ligands have the ability to activate effector immune cells that then, when activated, can destroy the cell that is presenting these ligands. There are also more sophisticated systems, and these have been shown today already, where immune cells can actually perceive what's going on at the DNA level if alterations at DNA level affect proteins. So these proteins, as part of their life cycle, they're processed, loaded onto HLA class I molecules, and then presented at the surface. And if you have a T cell that can recognize that aberrant protein, this would have the ability to delete the target cell. So we have grossly two different types of genomic instability in cancer. Either you have chromosomal instability, which is characterized by large chromosomal aberrations, or accumulation of mutations at nucleotide level, or so-called hypermutations. And those are alterations, of course, at nucleotide level. These go hand in hand in the majority of tumors, but you have few tumors like colorectal cancer or endometrial cancer where they're almost mutually exclusive. So you have some tumors that they're very prone to have chromosomal aberrations, but then they do not have a lot of nucleotide mutations. And you have tumors that have a lot of nucleotide mutations, but they do not have chromosomal instability. So if you think about which type of the mechanisms that I showed you before that would be more adequate to detect these sorts of genomic instability, I would say that the first one that I showed you is probably working best for the tumors that have aneuploidy, chromosomal instability, and have activation of DNA damage responses, while the accumulation of mutations, and if these occur in coding regions, that should be handled by the antigen-presenting machinery. And we have very good evidence that that's what occurs in colorectal cancer. So you have two types, the mismatch repair deficient and the polymerase mutant, which are hypermutated, and the mismatch repair proficient, where the majority shows chromosomal instability. And we showed that HLA class I defects occur primarily in the mismatch repair deficient tumors, the ones that have a lot of mutations at the nucleotide level. And of course, this is probably related to a selection of HLA class I aberrant phenotypes, and these tumors excuse themselves from showing aberrant proteins through the HLA antigen presenting machinery. This is also nicely correlated to the agent of selective pressure. So you see that the tumors that have lost HLA, you find more activated CD8 T cells in them. So you could link the phenotype with the agent of selective pressure. This is, of course, much more complicated than just an HLA T cell interaction, and there are, of course, other axes involved in this interaction. And it's even more complicated than this figure that I showed you here. It's not only a tumor cell T-cell interaction. For the sake of simplicity, I will restrict to this. So the positive signal that is received by a T-cell after the interaction with this T-cell receptor with a peptide HLA class I complex is counterbalanced with negative signals that are provided by other sort of interactions. So for a yang, you always have a yin and otherwise. So the interaction of a receptor like PD-1 on T cells with a ligand of PD-L1 gives a negative signal to the T cell that balances the positive signal by this interaction. And this is, in a simple manner, the explanation for the success of the anti-PD-1, PD-L1 therapies, that if you block this negative signal, you favor the activation of T cells and the elimination of target cells. So what has been seen, and others have already mentioned that today, is that there's a very high correlation between the mutation load in tumors and the response to anti-PD-1, PD-L1 therapies, which is easy to understand. The more mutations you have, the higher probability that a mutated peptide in a cell will undergo the antigen-presenting machinery and be presented to a T cell. There are other factors that appear to be correlated with the response to anti-PD-1 or PD-L1 therapy or other immune checkpoints, and those are pre-existent immune response. You do need to have a T cell there that is already recognizing an antigen in principle for anti-PD-1, PD-L1 therapy to work. It was already referred today, PD-1, PD-L1 expression is not a determinant for response to anti-PD-1 therapy, but it does seem to show correlation, particularly in some tumor types like lung cancer. And finally, a lot of groups have been associating the type of bugs that you have in your intestine, which are related to the development of your immune system, to responses to checkpoint blockade. So how does it look in colorectal cancer? Not surprisingly, after what I told you, the inhibition of this checkpoint blockade works primarily in mismatch repair deficient tumors. So the tumors that have a high mutational load. And you do not see any responses in mismatch repair proficient tumors, tumors with low mutation load. So the problem is this, is that the tumors that have high mutation load, or the majority of them in colorectal cancer, they already have a pretty decent prognosis. And that has been shown or has been known for a long time, particularly from the seminal paper from Gallon et al. So colorectal cancers that show a spontaneous, robust immune response perceived by the presence of T cells in the tumor tissues, they do much better. Their patients do much better than patients that do not show such type of immune response. And in our group, we're particularly interested in these and try to elicit immune responses in these patients. Of course, here the challenge is that there are not that many mutations to explore there. So the concept would be using the somatic mutations that occur in tumors which are coding, which lead to aberrant peptides, and choose those peptides to vaccinate the patients or to enrich for reactive T cells, give them back to the patients, and start a robust anti-tumor immune response. Well, a few years this was easier said than done. You have very few recurrent mutations between every patient. Most mutations are the so-called passengers, and they occur in a private manner. But luckily now we can sequence genomes in a couple of weeks, so it has become feasible to screen non-somatic mutations in a personalized setting. So what do? We get tumor and healthy tissue from the clinic. We perform DNA and RNA sequencing from the healthy and the tumor tissue. We're actually now also performing RNA sequencing from the healthy tissue. From there, we synthesize peptides corresponding to the non-synonymous mutations that we found. And in the meanwhile, we have collected tumor-infiltrating lymphocytes from the tumors and also lymphocytes derived from the peripheral blood. And then we test whether the peptides that we synthesize, whether they're able to elicit an immune response in the T cells that we have isolated from the same patient. If we do see an immune response, we have a very good argument to apply vaccination in colorectal cancer patients. So the probability that a non-synonymous mutation results in a neoantigen that is recognized in the T cells from the same patient is very low, at least from what we have seen published by groups like the ones of Rosenberg. So it ranges between under 1% to maybe 3% of the non-synonymous mutations resulting in neoantigens for which T cell responses can be detected. So we gave it a thought, and we were wondering whether we were doing our best to detect the maximum amount of non-synonymous mutations in a given tumor. As you know, one of the crucial steps to detect mutations is related to exome capture, so that you focus on coding regions, and only then you do the sequencing. And when we looked at the exome capture that is being performed by most groups, we noticed there is a very irregular coverage of the coding region by sequencing reads. And this was very striking when, for the same sample, we produced more sequencing reads, but we couldn't increase the amount of coding genome that was being covered by those reads. So I guess a lot of us has the experience that when you strict to the coding region, you will have, to the coding genome, you will have more or less 80-something% that is covered by at least 20 reads. And you need quite a lot more than that if you want to detect non-synonymous mutations in a sample that is not pure, and also to detect subclonal mutations. If you see, when generating 50% more reads for this particular sample, you do not really increase the proportion of your coding genome that is covered by a decent number of reads. So we started testing a number of different captors in the lab, and one of those was the one detected or developed by IDT, and it was striking after performing the alignment how smooth the coverage of the exome was. And it was even more striking when we went again and looked at the depth of coverage data, where you see that nearly 97% of the coding genome was covered by at least 30 reads. And this is quite constant if you go further up. And when comparing the previous captures that we were doing in terms of its ability to detect non-synonymous mutations, we did indeed increase the number of non-synonymous mutations we detected by about 10%. But as in any method, you also then miss something that was detected by the previous capture. But when we look closely, these 32 non-synonymous mutations that were not detected by the new capture method, they were just not designed to be targeted, because they derive from rather obscure transcripts for which it's not known whether there's actually a protein. So the new capture method is working pretty nicely. So for this specific case, after optimizing the detection workflow, we ended up with 600 non-synonymous somatic mutations. And one of the filters we have been employing is to check whether then these non-synonymous mutations are expressed at the RNA level. And we also sat together and wonder whether this was being done correctly. There is one problem with using a sequencing strategy to detect RNA expression, is that you have a limited number of reads. So let's say if you only have a number of, or 10 reads available or 100 reads available, and you want to assess the expression of 3 genes that are separated in levels of expression by 10-fold, you will not be able to detect the expression of gene 1, although it might be expressed, just because you don't have enough sequencing reads to get it. So when people always say that this gene is not expressed, you should always correct and say it's just not detected. You cannot assess whether it's not expressed or not. So we wanted to see from how many non-synonymous somatic mutations there was actually RNA in the cells that was not being detected by total RNA sequencing. And for that, we did amplicon RNA or cDNA sequencing. So we designed M13-tailed libraries around the non-synonymous mutations for this patient. And what we saw is that 1/3 of the variants that was not detected by RNA sequencing were indeed present in the cells. They were just not detected by the total RNA sequencing approach. So you may wonder whether RNA sequencing is necessary at all. It is because, for instance, for splicing mutations, we do not know what is the consequence. So we need RNA sequencing to know where the new splice sites reside, which might generate new epitopes and might include even frameshift mutations. Also for the detection of RNA sequencing fusions, if these give rise to proteins, that can also be called a neoantigen or defined as a neoantigen. And it's very useful to have RNA sequencing data as well. If you do not have HLA haplotyping data on a patient, that can easily be done with RNA sequencing, and also to detect HLA-specific or allele-specific HLA class I losses, which will have an effect later when you predict which neoantigens you will use to give to a certain patient. So what is the overall result of all this work? So for the patient I showed you before, when we put everything together, when we predicted by net MHC which peptides might bind the HLA alleles from these patients or haplotype from these patients, we increased the number of peptides that are going to be tested in functional analysis by 50%. So I think that's very relevant if you take into account the number of neoantigens that are detected from the total number of non-synonymous somatic mutations. I still have some time, or should I stop here? I can also stop here. No. So often people say or ask, so is it feasible to do neoantigen vaccination when you already have advanced tumors? And I would say yes, and there's enough evidence for that. And I just selected the seminal case from Rosenberg's lab, where there was a cholangiocarcinoma patient with metastatic disease, and a tumor sample were sequenced for exome and RNA sequencing. Neoantigens were produced from only a total of 32, and a T cell was identified, or a T cell clone was identified that recognized one of those neoantigens. These T cells were proliferated to great numbers, given back to the patient, where it was shown that the tumor regressed in 2 different occasions after 2 different infusions of a T cell product. We also recently shown that T cell responses are still occurring even though a patient already has widespread disease. And this was a work that we published last year of a melanoma patient, stage 4 patient with metastatic disease that had been treated by adoptive T cell transfer. So the metastases in 2005 were collected from this patient, and those metastases underwent exome and RNA sequencing from which neoantigens were identified. And these were tested against the T cell product that was given back to the patient. And this T cell product was obtained by incubating lymphocytes from the peripheral blood from the patient with a cell line that had been generated in 2005. And what we saw is that the T cell product contained T cells that recognized 2 mutations present in the KAIA and the RPL28 gene. This patient underwent another resection in 2006, and then we did the same exercise, but then we looked at the tumor-infiltrating lymphocytes that were in the tumor tissue in 2006, and there we only detected responses against the KAIA protein, but not anymore against the RPL28. And by next-generation sequencing and then validation by Sanger sequencing, which is easy to demonstrate, you saw that the clone that contained the RPL28 mutation had disappeared. And although we cannot claim that this was an immune-mediated reaction, it's very well conceivable or obvious that that was the case. So I would like to conclude that for the detection of maximum amount of potential neoantigens, optimal genomic approaches are warranted. And people really have to consider whether they just want to have a service that provides mutations, a mutation list that they will then, produce and test in the lab, or if they want to collaborate with labs that actually have genomic expertise and can work or can be dedicated to your project and come up with the right solutions. And I hope to have showed you, or to convince you with a few data that I showed you, that even in the case of metastatic disease, antigen-driven immunotherapeutic intervention is possible. And I will end here by acknowledging my 2 PhD students, my all-round technician, Nechud, and Dina, the bioinformatician, and the funding sources. Thank you.